Hello, everyone. My name is Paul from Edgene. It's very nice to meeting you. Edgene is a biotech company pioneering the future of mitochondrial genome editing. We have platform technology for mitochondrial genome editing. The problem is mitochondrial genetic disorders. It's maternally inherited disease. It affects 1 in 5,000 individuals. Mostly, it caused by single point mutations in mitochondrial DNA. And there's no cure at all so far. That's why patient face progressively vision loss or organ failure, even death. Why there is no solution so far? Because CRISPR, well-known genome editing technology, cannot into the mitochondria because of RNA structures. And also, traditional TALEN, that's the DNA cutting method, is cause massive cell death rather than repairs. And also, in vivo delivery required to target organ is have some kind of barriers today. And Edgene has solutions. We have only solutions. We have a CRISPR-free base editing technology. Edgene's proprietary base editing platform bypassed the guide RNA delivery barriers, enabling direct and precise repair of mitochondrial DNA mutations with surgical accuracy and minimum cell death. Our lead asset, EGR104 for LHON. LHON causes sudden blindness in young adults. EGR104 target is ND4 gene in mitochondria mutation, especially 11778 position of nucleotide, G2A mutations. We have nice adenine base editors, and we can cure those LHON patients with ND4 mutations. Our groundbreaking data in this is surrogate animal model data. We produced mouse model of ND4 gene mutations in mitochondrial DNA using cytosine base editors, and we designed adenine base editors that can cure those mutations. It converted to the mutated form to wild types. And we published in Nature Communication last November, and one shot our asset, not our asset, it's a surrogate model. One shot of gene therapy asset can recover the function of eye, also recovery of retinal ganglion cells. Our Edgene has a world first and best technology for adenine base editor, so-called TALET, and cytosine base editor, so-called Hi-Fi DDCB editing technology. We covered most 84% of mitochondrial DNA point mutation can convert to the wild types, and with high fidelity. That means our technology can apply to human therapeutics. And we have a strong global IP portfolio, and our core platform technology and improved one filed in 13 countries, registered in now 3 countries. We are going to more registered. And also we have pipeline IP for our assets, and we are now proud to be, we are only mitochondrial genetic disease can cure by our technology. The market is massive. The gene editing market is will increase, and around 13 billion US dollar by 2035, and mitochondrial disease sector is converted to over 2 billions. Our company is a first-in-class for the mitochondrial DNA mutation therapies, and we can overcome those kind of markets. And you see some our competitors in Pretzel or Gencyte or Primera in US, but those kind of the company has traditional gene replacement therapy or unknown modalities. Edgene is standalone for mitochondrial DNA, the therapeutic gene editing technologies. Our company is led by the founder and CTO, Jin-Soo Kim. He is a renowned scientist in CRISPR area. Now he invented mitochondrial DNA-based editing technology, and he found Edgene, the company Edgene, and me myself, the 25 years in drug development in biotechnology area, and we together and elite R&D teams in Edgene, we can make executions. We have nice ecosystems from academic innovation through advanced delivery or targeted therapy areas. And our roadmap is clear, the milestone driven. We got technology optimization through the year 2025, and preclinical studies candidate selection in these years, and we progressed to the GLP tox studies by 2027, and finally we can phase 1 try entry in year 2028. And our business model is our licensing, early stage licensing to global big pharma, as like Pfizer also. And US FDA first tactic is we can make money from the rare disease. And also we expanding to our technology to chronic disease as like Parkinson's. And now we are the fundraising for the mature our pipeline, 20 million US dollar Series A, and we spend most of the capital, the 75% of capital to CMC and IND enabling studies to mature our pipeline to enter the clinical trials. Our company Edgene wanted to cure the incurables using our technology, direct mitochondrial genome. Yeah, that's all. Thank you for your time.
When you look at the vertical transmission, are you able to monitor any of the offsprings?
Actually, we do have some animal models, but we don't see any the mitochondrial base change can into the offspring, right? Yeah.
Yes, that you can monitor that.
Yes.
Okay. And the second question is the delivery. How are you delivering into mitochondria?
Yeah, in the case of LHON, our target is retinal ganglion cells in eye, and we are now thinking about adeno-associated virus for gene delivery of our asset.
Okay, thank you.
Any other questions? By the way, if there's any questions online, folks online, if you have any questions for the speakers here, feel free to type it in the chat or just simply unmute yourself. I think we're a small enough group here. So if you have any questions, feel free to unmute yourself as well. Okay.
Okay, thank you very much.
Thank you.